Now, even though the blood and the CSF have different solute compositions, there's still a really close relationship between those two extracellular fluid compartments. And that's because the CSF is formed from the blood. So the blood supply to the brain is mostly consisting of the carotid artery, which branches out to supply the anterior and the medial portion of the brain, and due to the vertebral artery, which supplies the posterior structures of the brain. Now remember that nervous tissue has very, very high requirements for oxygen and ATP because of all those active transporters. So it doesn't tolerate interruptions to blood flow very well. So one helpful specialization inside the brain is the circle of Willis, which creates redundant or overlapping blood flow. You can see the circle of Willis at the center of the diagram on the left-hand side. And the idea is that basically by having the circular type of arrangement of the vessels, if there's a blockage or an interruption of blood flow on one side, then blood can reroute and go the other direction and still make it out to the tissues. But the circle of Willis can only protect you against blockages that are taking place inside the brain. So if there's an interruption to blood flow earlier in the pipeline, this can still cause pretty severe problems, including things like stroke. But this is one of the reasons why there's a lot of controversy around the use of force and specifically around chokeholds. Now, chokeholds typically compress the carotid artery, and so they stop blood flow before it gets to the circle of Willis. This deprives the brain of glucose, which it needs to drive those active transporters, but more urgently, it also deprives the brain of oxygen. Doing this even for a few seconds can lead to confusion, disorientation, and cognitive impairment, and eventually it can cause someone to go unconscious. But it can also very quickly lead to death, as we saw most recently in the killing of George Floyd. Now, even in situations where it doesn't lead to death, oxygen deprivation can cause irreparable ischemic injuries to the brain. Ischemia is a general term that we use to imply damage due to oxygen deprivation. But this is the reason why under normal circumstances, about a fifth of the body's blood is going to the brain at any given point. If you look at the table on the right-hand side of this slide, again, you can see sort of counterintuitively that the glucose concentration in the CSF is actually lower than the concentration in the plasma. But that's actually because the neurons are constantly consuming glucose. And so therefore, glucose is continuously being transported out of the CSF into the cells. And that keeps the concentration in the CSF a little bit lower. So counterintuitive, but also makes sense if we think about what's actually happening with the tissue. Now, pretty much anywhere that there's a blood vessel inside the brain, you have a blood-brain barrier. And that allows the difference in composition between those two compartments to be maintained. The regions of the blood outside of the choroid plexus produce the other 25% or so of the CSF. So about 75% from the ventricles and about 25% from other places. The blood-brain barrier is formed primarily from endothelial cells. Those are epithelial cells that form blood vessels. They're represented in red in this diagram. They have a lot of tight junction proteins between them, so that limits paracellular transport, and it makes the exchange between the blood and the CSF extremely selective because it has to pass through those cell membranes. That's why it's called the blood-brain barrier. The selectivity also helps to prevent the leakage of ions and neurotransmitters. It keeps out toxins and it keeps out pathogens that might be in the blood. So thinking one final time about the difference in composition between CSF and the plasma, one of the reasons why the composition of protein in the CSF is lower than the blood is to avoid giving pathogens the building blocks that they would need to replicate within the brain. By keeping the amino acid concentration low, for example, you can keep viruses from replicating. Now, the tight junction proteins in large part are regulated by a supporting cell known as astrocytes. Astrocytes are a type of glial cell. Astrocytes can signal to the endothelial cells when there's a need to increase or decrease the expression of tight junction proteins. And so, therefore, they can regulate the exchange. In addition to tight junctions, the blood-brain barrier is also regulated by regulating blood flow. This is due to a type of contractile cell called a pericyte. When pericytes contract, they can reduce the blood flow and therefore limit the amount of exchange that can happen. Now, collectively, all of these cells that I've just mentioned that regulate the blood-brain barrier are known as a neurovascular unit, or NVU.